go to the 1:30కి సెక్షన్స్ రెడీగానే ఉన్నారా? ఓకే సార్. లేట్ అయింది సార్, 1:30కి షార్ప్ స్టార్ట్ చేయాలి. ఓకే సార్, అంటే ఒకసారి రిమైండ్ చేయడానికి కాల్ చేశాను సార్. సార్ యూ కెన్ కాల్ మీ రాత్రి, డోంట్ నీడ్ టు కాల్ మీ. ఓకే సార్, షూర్. వచ్చేస్తాను సరే సార్, కాల్ చేయండి సార్ ఏదైనా డిస్కంఫర్ట్ వస్తుంది.
Srinivas, good afternoon.
Hello, Srinivas sir.
Good afternoon. Good afternoon. Good afternoon.
Okay. Thank you, sir.
How are you, sir?
Fine, Andy. So, how is the voice? Is okay, kada?
Anta okay, sir. Everything fine.
Okay.
So, good afternoon to all the delegates. Welcome back to the section 3 of day 5. Myself, Lakshman Vinod Kumar, Assistant Professor, Department of Pharmaceutics, VV Institute of Pharmaceutical Sciences, Goodlavallaru. It's my great honor to act as a chairperson for this section. Now it's time to introduce the resource person of this section, Dr. Srinivas Nanduri, Professor in Naipur, Hyderabad. Sir completed his BSc in 1986, MSc Chemistry in 1988, and he got his PhD in Chemistry 1991 from Sri Satyasai Institute of Higher Learning, Prashanthi Nilayam. Sir accomplished medicinal, synthetic, and natural product organic chemist with 30 years of industrial and academic research experience. associated with several Indian companies like Indian Pharma, CRO, Dr. Reddy's Laboratories, Biocon BMS Research Center, Albany Molecular Research Center, Sri Satya Sai Institute of Higher Learning, National Institute of Pharmaceutical Education and Research, Hyderabad, and reputed academic institutions. SAR played different roles like lecturer, senior director, chief scientific manager, research director, director, associate professor in various reputed organizations. SAR acted as a dean in NIPER, Hyderabad from March 2020 to September 21. Now, SAR acts as a professor in Department of Chemical Sciences at NIPER, Hyderabad from December 2019. SAR guided more than 50 MS Pharma and MTech Pharma students and also guided 5 PhDs, and he has 10 ongoing PhDs in his credits and led to several integrated drug discovery programs encompassing all stages of drug discovery like hit generation, hit to lead, and lead optimization involving both natural product and synthetic products in the areas of oncology, metabolic disorders, and various infectious diseases. SAR got funding research from Ministry of Earth Sciences, which is worth of 45 lakhs as principal investigator, and from DST SCRB, which is worth of 43 lakhs as a principal investigator, from DBT, which is worth of 40 lakhs as co-principal investigator, and also from ICMR, which is worth of 50 lakhs as a principal investigator, and from LSRB, which is worth of 49 lakhs as a co-principal investigator. SAR also developed 4 research labs for the chemical synthesis. SAR published 57 research articles in peer-reviewed journals and filed 54 patients, in which 19 are granted. SAR acts as a speaker for more than 30 conferences and organized 13 national and international conferences as convener. SAR also undertake different responsibilities like coordinator, National Center for R&D Bulk Drugs Development, coordinator, Avishkaran Bionest Incubation Center at NIPER, Hyderabad. With this brief introduction, I hand over the session to resource person, Professor N. Srinivas sir, who give a talk on applications of artificial intelligence in drug discovery and development. Now the session is all yours, sir. Thank you. Over to Srinivas, sir.
So, thank you very much for your kind introduction.
Thank you, sir.
And good afternoon to everybody. So I think as it is an afternoon session, I will try to make it as interesting as possible so that all of you will maybe already 3 days have passed. This is the 3rd day. That's what my understanding. So I think it will be I'll try to give more of an applications of what we have done, especially the one which I have taken the application from my own work, so that it will be much more interesting for all of you. I will share the presentation. So can all of you see the slides, please?
Yes, sir. Yes, sir.
Yeah. Okay.
You can continue, sir.
Yeah. So my request is, whenever your the time or and whenever if you are facing any issues with my presentation, just please alert me, and then also just alert me on the time also, so that I will not be crossing the time. Okay. So the title is very aptly put it like artificial intelligence and drug discovery. So where we have actually there are two sentences, two words called, one is artificial intelligence and another is called drug discovery. So when we talk about drug discovery, I think all of you know that it is a very long process, and it has to be, and it's a process which actually has many challenges, and also many times it so happens that the success rate may not be very high. So that results into high cost of development. And then what is important is artificial intelligence. And when we talk about this artificial intelligence, so that's actually intelligence is actually originally it is a human intelligence, and it has to be artificial intelligence is nothing but using the machine tools for as a substitute for human intelligence, so that we can make much more accurate decisions regarding to the drug discovery. But my experience and my intuition and my not only my experience but everybody's experience says that artificial intelligence cannot replace completely the human intelligence. Because that is something, human intelligence is always a superior one, and it is a tool in the hands of human to accelerate the drug discovery process. And also many times, there are some places where actually we humans can err. So using the machines, maybe, we'll try to minimize that errors, that, to reduce the failure rates. So that is the only the advantages of this artificial intelligence. But I think a good combination of human as well as artificial intelligence is very much required for a very complex program, very complex process like a drug discovery. And then now going to the what is actually the artificial intelligence. Then here what we are having is, this John McCarthy, who is considered as a father of artificial intelligence. So here's the definition is the science and engineering of making intelligent machines. So what actually he means is by the artificial intelligence is by the intelligent machines, the ones which actually can make decisions on behalf of humans, and that is why they are called intelligent machines. And what is called the science and engineering of making intelligent machines is something which is called as a artificial intelligence. Artificial intelligence refers to the ability of a computer or a computer-enabled robotic systems to process information and produce outcomes in a manner similar to the thought process of human in learning, decision making, and solving problems. I think as we see this sentence, it is not artificial intelligence. It's just not that, the one which has just come up, propped up in the last few years. It has been there in our system, and actually the what we are using the computer enabled systems or computers processes for processing and producing the outcomes which are similar to the thought process of the human being learning. And the goals of AI system is to develop system capable of taking complex problems in ways similar to human logic and reasoning. So this is what we call it as a artificial intelligence. The deep learning, what is actually is coming again from the machine learning. Deep learning is a subset of machine learning where artificial neural networks, algorithms inspired the human brain, learn from the large amounts of data. So this is something that and where machines can do a lot of different things, where we have the large amounts of data. For example, let us say a drug which is actually already in the clinical trials. Let us say it is subjected to so many, it is subjected to the treatment of many either normal human beings like a phase 1 studies or phase 2 studies wherein you are working on the drug is given for a patients, and the phase 3 studies where you have a large set of patients, and then the data which is derived from the patients in terms of the clinical data, to that's a huge amount of data. And how do you process that? That is very, very important. And then so to assess whether whether the drug is a safe drug or whether the drug is having the efficacy which is enabled, which is required. So all these have to be taken into account. And for processing that much of complex data and also huge amount of data, requires the machines to help in doing that. Then what is actually the applications of pharmaceuticals? Applications of artificial intelligence in pharmaceuticals. And if you really see this cartoon, I think there is no area in pharmaceuticals which is where the artificial intelligence has not played a role. So for example, you take the drug discovery process. You have a drug design wherein you talk about this target protein structure, drug protein interactions, de novo drug design, all the structure based drug design actually is completely is done by the artificial intelligence. And the tools which we have used, like the molecular modeling, molecular docking, so virtual screening, all of them they come into the drug design part. The second part is what is called the drug screening, wherein actually the drugs are evaluated again for their bioactivity and the toxicity and physicochemical properties. And I think all of you are aware that there are a number of tools, a number of software available for for predictive softwares. Most of these called ADME predictors, and these things come as a drug screening. Then the third aspect is actually what is called as a pharmaceutical product development. So when we say it's a pharmaceutical product development, so that is nothing but final drug, so or the finished dosage form, where is actually a new formulation is done, wherein you have to talk about you have to make a decision about what kind of excipients are required, how do you monitor and modify developing processes, and ensuring in-process specification compliances. So these are the three aspects which go into the pharmaceutical product development. That is also applications of artificial intelligence are there. Then after the drug discovery, then formulations, then you are going to the manufacturing. Manufacturing right now, it is more focused on the automated manufacturing, so that it can be much more programmed and which can be done. And this personalized manufacturing, where you require to do it's a customized kind of a manufacturing, and correlating manufacturing errors to set parameters. So this is again a where machines can play a very important role. Then finally, the QA and QC part, which is actually playing a very, very important role in the current scenario, where regulatory compliances are very stringent, and then often it requires a very vigorous process. So the rigorous process involves understanding the critical process parameters, guide future production cycle, regulation of inline quality, ensuring quality assurance with aid of electronic blend motifs and other techniques. So this is something. Then the last one is subjected to the clinical trials, when when the molecule has gone through from discovery to the manufacturing, then QA QC, then enrollment of various human subjects, then looking at the patients who are dropping out, monitoring of the trials, then selection of collection of the data, then processing of the data. So the whole thing actually, this is where actually the large amount of the data of the patients is monitored, and also it is monitored, and also it is actually the analyzed. Then finally, when it comes to the product comes to the market, so pharmaceutical product management, where you're thinking of market positioning, market prediction, analysis, etc. So these come into the picture. If you really see the whole cycle of drug discovery to the commercialization, so artificial intelligence is playing a very major role. And what actually kind of a suitability of deep learning for drug discovery? So if you really see the theoretical aspects, so expressiveness is something neural nets can approximate any function. So that means the ones which are present in the artificial intelligence, they can approximate any function, and then applicable to many domains. Neural nets are trained easily, so that means you can program any of the generalizability. Neural nets generalize surprisingly well to unseen data, and practically what we see is applicable to many domains. It is a hard to deep learning can it's hard to beat as long as there is no data to train, and scalability is better with more data, and it is very scalable. So if you really see that deep learning or the machine learning or the artificial intelligence, they are not only from the point of view of theory, but even from the experience point of view, they are found to be more general, very competitive and scalable. So hence this is actually the applications of the artificial intelligence are making more and more inroads into the drug discovery process. So when we talk about the drug discovery, so drug discovery is actually a very critical one. It is a process through which potential new medicines are identified or discovered. It involves a wide range of scientific disciplines, like including biology. So then you have a chemistry, so molecule people making a lot of chemistry molecules, and you have biology, where we talk about the various drugs, and also when we talk about these two putting together, bound drug target co-complex. So this is very important to note. And all of you are aware of this. So it starts with a target to investigation a new drug. It goes through hit design, hit to lead, and lead optimization, preclinical development, and these are some of the kind of parameters which are given in each and every one. Then, as you as you are aware that the nature of drug invention is very, it is a high cost and low success. The title may look very not optimistic, it is a pessimistic kind of a thing, but it is a fact to note. So maybe I would say that even though it's a high cost and low success, but I think that it is worth doing the process, because there are many which are actually discovered and then marketed, and which are and if you really see many of the drug discovery which has happened now, those is really coming to they are helping a lot the patients. Supposing if you really see in the coming in the current scenario, I think many of the diseases, they have been successfully supplemented with new and efficacious drugs, and they are very safe. They are found to be very safe. They are very successful drugs. For example, let us say you take the example of JAK inhibitors for inflammation. It is a very successful drug. So all the JAK inhibitors have been very found to be very successful for treating the rheumatoid arthritis. And like I say, similarly, you are having a large number of chemotherapeutic drugs for treating the cancer. And you take, I think, namely, you take the hypertension, you take metabolic disorders, you take diabetes. So I think every disease, there are successful drugs, and all through, even though the drug discovery process is very stringent, but and also it is wrought with high cost and low success, but still it is worth doing it. And also many industries have been pursuing it very passionately, even though they have been found to be having low success. And I think it's a high risk reward game. So you start with a drug discovery, and you start with the 10,000, more than 10,000 compounds, then preclinical development to all the way to FDA approved. So if you see from 10,000 to 1, it looks a very kind of a very dim view, but I think the end there, if you are having a drug which is actually a which makes a company, and then which and also touches a lot of patients' lives. And what are the three basic questions in discovery? So given a molecule, first you have to find out whether the molecule is a drug-like properties it is having, and whether it can modulate which drugs targets, and then it's what kind of a dynamics, kinetics, effects, metabolism if administered orally or via injection. Given a target, supposing if you are having a target, what kind of a molecules can act on the target, and among the number of molecules which are acting on the drug, pick the good one. And if evaluation is expensive, need to search. And if no molecule is found, need to generate from the scratch. Then how does the drug-like space look like? Supposing now this is a scenario. For example, let us say first you have a molecule, especially when we talk about drug repurposing. You are having a molecule and you are trying to find what kind of activities it can do. The second is you have a target, but you do not know what kind of a ligand it can make. And this is where actually a lot of innovation can happen. And then steps to make molecules. If you know target and if you know what kind of molecules, then how do you make molecules? Then this is, I think all of you are aware of this, what is called as a virtual screening. It is a computational approach to assess the interaction of an in silico library of small molecules and the structure of a target molecule to rapidly identify new drug leads. So you have a merits computational, only high scoring ligands goes to assay, demerits molecular complexity, diversity, false positive, synthetic issues. Then a typical workflow of a virtual screening. So you decide a lot of large chemical libraries or QSAR modeling. Then you have a targets which are given here, and then go through the virtual screening. Then finally synthesizing the prioritized compound. Then it goes to in vitro assays, in vivo assays, it to lead and lead optimization. Then it goes to the laboratory collections and literature data. Again, it comes back to this process. So this is a typical workflow, and actually I will be demonstrating this in one of the case studies. So molecular modeling, I think all of you are aware that there is one of the virtual screening methods. where actually it's a method which predicts the preferred orientation of one ligand. So when you know a target is there, and you know whether the ligand which is actually is expected to whether the it will actually give a very quick idea of whether the ligand which we are thinking, whether that will really bind to the desired target or not. So, you know, for that actually is what is called as a molecular docking. And you know there are two types of a docking. One is called rigid docking, and another is called flexible docking. So flexible rigid docking, you know, it's a the internal geometry of both the receptor as well as the ligand are treated as rigid. But whereas in the case of flexible docking, there is a good possibility of flexibility, especially in the active site. So I want to take you through one case study, which in which actually I was involved as a principal investigator, and you know the molecule, you know, we were at the end of it, we are able to make a molecule which is currently in the phase 3 clinical trials, which is as a medicinal chemist, it is a very successful program from my side. I just want to take you through, and you know the idea of taking this case study is not that, you know, we are having completely the whole data is generated from the artificial intelligence or the machine learning, but it is a good combination of the artificial intelligence techniques, or especially, you know, computational techniques, at the right opportunity to the right direction for this in this project. So the project is talking about, you know, one of the protein kinase inhibitors. I think all of you know that, you know, protein kinases, they play a very significant role in cell signaling and implications in human pathologies. And when we talk about cancer, it's a normal cells where activation of protein kinases leads to cell growth and differentiation. And then when it is deregulated, so the tumors, so the activation of protein kinases, it actually leads to overstimulation of cell growth, and the activation of protein kinases suppression of apoptosis. So it leads to, in a tumor cells, it is uncontrolled cell proliferation and survival of apoptosis. This is something, a major difference between a normal cell and a tumor cell. And protein kinases, what do? They do the transfer of the phosphatase from the ATP from the ATP and becomes ADP, and then, you know, it actually phosphorylates the phosphoprotein. So for example, in this case, if this is the target therapeutic target, so that means, you know, all the, as you know, that all the therapeutic targets are more or less, they are having a polypeptides. It's a small enzymes which are nothing but a peptides. And in this, when we have a phosphorate transferase inhibitor, so, you know, when actually this phosphorylation happens, and it's similar, for example, like, you know, this tyrosine, you have OH, it getting phosphorylated, and the ATP in the process, it transfers the phosphatase, and then it gives rise to the it becomes the ADP. And you know that, you know, there are in a cell we have a number of regulatory growth factors. The growth factors could be negative factors or positive growth factors. Positive growth factors leads to some kind of a cellular responses, like, you know, cell proliferation or cell differentiation. So whether it is a negative or, so you have it goes through signal transduction, and then actually it triggers the nucleus to, and then, you know, there are many responsive cellular events which happens. And what we are talking about, and what I am going to discuss about, is there is a receptor called fibroblast growth factor receptor. I think all of you know that, you know, the growth factors, like a epidermal growth factor is there. So similar to epidermal growth factor, fibroblast growth factor is one more very important growth factor which is actually present on the cell surface. So you can see here in this cartoon, you can see there is an FGFR here. So in this FGFR, you are having FGF is a ligand, and this is the receptor. And the FGF, that is nothing but the is a polypeptide growth factors. It's a 22 member family which shares 120 amino acids. It contains around 120 amino acids. FGF receptor binds FGF binds one of the subtypes of FGFR inducing tyrosine kinase inhibitor. This is a tyrosine kinase inhibitor activity, tyrosine kinase activity, and transmit the signal by sequential phosphorylation of downstream processing. So you have the sequence of events which actually happens to intracellular functions. And what kind of functions it will have? It will have a angiogenesis and differentiation. And many, when this whole process is any at any given process of this is deregulated, then it leads to several tumors or several deformed kind of a one, and the one which is more expressed is in the cancer. For example, when the FGF1, FGFR1, which is one of the receptors is FGFR1. If the FGFR1 is deregulated, it is actually leads to brain, breast, prostate cancers. FGFR2 is a gastric, breast, endometrial, small lung cancer. FGFR3 bladder cancer, endometrial, myeloma. FGFR4 prostate, breast, colon, melanoma, lung and human and neck sarcoma. FGF's increased expression of tumors. FGF2 to FGFR1, it leads to angiogenesis. So these are the this is a promising target, and actually it's the besides the epidermal growth factor. So this is something which is a very challenging target to work with. And at the time when we took this project in our hands, there are molecules which are given like from different companies like Bristol Myers Squibb, Martis, ESI. Then you have Boehringer Ingelheim, then Bayer. So you have a number of companies which are working on this. But what we notice is most of them not only with the given targets like FGFR, but they are also targeting many other kinases. Like for example, if you take the Novartis molecule called dovatinib, so FGFR, PDGFR, VEGFR, FLT3, c-KIT. Then supposing if you say GlaxoSmithKline GSK, so there is also very similar to this. Then you see, for example, Entramed. So you have more than several kinases in which actually this is effectively targeting. So in a kinase inhibitor, actually the major challenge is to get a molecule which is of the very selective in nature. So that means it is supposed to be, it is actually expected to the selectively inhibit the desired target, but not all targets. Actually, the multi-kinase inhibitors are considered to be, expected to be much more toxic compared to the selective targets. So that is why in the kinase inhibitors are always, if aim is to get a selective molecules. Then when we started a program, so our aim is to identify potent and selective inhibitors of FGFR. And then when you say potent and selective, but also being in a commercial organization, it should be identify patentable lead molecules. So that is very important. Unless you have a patentable drug, you will not be able to get a the commercial success. Then you have to demonstrate proof of concept in appropriate disease models, because that is as that before we go to take the molecule to the clinicals, so you need to do a lot of preclinical studies in which actually the disease models are nothing but the animal models. And then to optimize lead compounds and bring a development candidate to the regulatory toxicity testing stage. So this was the objective with which we have started working. What are the challenges we have? So I think this is where actually many of you will appreciate how we are going to take up a drug discovery program. And whenever we talk about drug discovery, it is not a simply artificial intelligence directed program. It is actually a combination of human intelligence and artificial intelligence, where we have to judiciously use the artificial intelligence tools, like at appropriate places to take the program to the forward. Absence of co-crystal. What we had the challenge was we did not have FGFR, so which is co-crystallized with the ligand. So that was the challenge we had at that time. And then since when we started, it was a very we had only multi-kinase inhibitors, but not selective FGFR inhibitors. There is no chemical starting points actually where we could start with. So this is the challenge. So what we have done in our thing is we screened all our chemical libraries which are present in molecules. Then we had taken a FGFR overexpressed it, and then we have standardized a biochemical assay of FGFR. Then what we have done is at a 1 macromolar to 10 macromolar. So we have screened all of molecules, and then what we found is we found a number of benzimidazole molecule. I think all of you are aware that this is a benzimidazole core. So this benzimidazole core with a N-phenyl here. So this one is actually a very promising hit. Then what we realized is where the one which actually had a molecule like this. So this, but of course, I should just give you a background here. More than 100 benzimidazole and benzimidazole-like compounds, benzothiazoles, benzoxazoles we have evaluated. Out of which, what we have found is we have found that compound 1. So this compound 1, if you really carefully see, you have a benzimidazole here and you have an N-phenyl here. And in this N-phenyl, there is a meta position here. I think you are having a N-acetyl moiety. And on this side, you are having a substitution at this position. So that means if you see start from 1, 2, 3, 4, 5, 6, actually 6 position, you are having a substitution. What kind of substitution here? It is a pyridine substitution. Then when we have screened the molecule at on FGFR1, at 1 macromolar, it has shown 38% inhibition, and 100 nanomolar, it has shown 8% inhibition. And then this actually on then we had another molecule called compound 2. The difference between compound 1 and compound 2 is this position here is not substituted, but whereas alternatively this is substituted here. But this also has a benzimidazole and N-phenyl, and at a meta position you are having N-acetyl, and at this position instead of having a meta pyridyl, so it is a para. So this compound 2, if you really see the difference between this and this, FGFR1, it is at 100 nanomolar 69% inhibition, and 1 macromolar it is 86% inhibition, whereas when we calculated IC50, it is almost like a 30 nanomolar. So you see the drastic difference between compound and compound 2 just by the change of position of this substitution here. It changed a lot, and this became much more active. So now this is actually the starting point what we had. But when we analyzed this molecule, what we found is when we put it into, when we get a molecule, then the immediate thing we have to see is whether, let us say, you are already having a molecule with 30 nano potency. So the molecule is put into the various, first to see whether the molecule is metabolically stable or not. I think all of you know that metabolically, to study the metabolic stability, you use human liver macrosomes or mice macrosomes, rat liver macrosomes. So these are all the in vitro studies, and also when you do a simple PK studies, also you see whether the molecule is stable or not. What we found is the molecule is unstable, so metabolically. So that means this molecule can be susceptible for metabolic transformations. Then what we analyzed is there is a N-acetyl moiety here. That could be one of the challenges. You are having a 4-pyridyl, so wherein this nitrogen is actually exposed. So when it is exposed, what happens is this becomes a N-oxide. So that is something which has to be blocked. Then what we changed is we did not immediately work on the N-acetyl, but what we changed is pyridyl we have changed it to N-methyl pyrazole. Now this change of phenyl to pyridyl to N-methyl pyrazole, this has also improved IC50. It has become 13 to 18 nanomolar. Besides that, this molecule is quite stable, and also when we have better metabolism, it's stable, and actually when we have done it has a good oral bioavailability, and then a preliminary in vivo studies has shown that this molecule is having a promising in vivo activity. So that means what we are able to do in this cartoon actually, which I could capture almost like, maybe 6 months to 1 year of our work, wherein we have screened a large number of benzene molecule. What we are able to show is from virtually inactive molecule, we could optimize to get a compound like compound 3, which is having a very good metabolic stability, oral bioavailability, and also bioactivity in xenograph studies. So this is something which is a great, it is one of the good steps which actually in the directions. But what is
Excuse
it?
me,
That
sir.
is it really a good drug-like molecule? Is it a does it qualify to be a drug? So this is a challenge which we have to uh, which we try to answer it ourselves. But what we found is when we know that the molecule is acting on a kinase like a FGFR1, so we know that we are having a we have to show that whether the molecule is selective towards the desired target, or whether it is whether it is unselectively, or whether it is affecting everything. So for example, if you take the TK, it's called as a dobutamine from the Novartis that we have using as a starting as a standard prophylaxis. So here, this molecule, which is as
Sir, you are not audible, sir. Hello, sir.
Sir, your mic got muted. Please unmute yourself, sir. Srinivas, sir. Srinivas, sir, please unmute yourself.
so you could hear till now, or
Yes, sir. Yes, sir.
okay, only just now it became right. Hello,
You are audible,
hello,
sir.
can you hear?
You can
Now
continue, sir.
I can continue, right? Okay, okay, thank you. So we have we have the selectivity profile. Then at 1 micromolar, what we found is this compound 3 is actually hitting or inhibiting aurora kinase. Aurora kinase is actually, again, it's one of the very promising. It was considered as one of the very promising kinases for in the cancer. And however, later actually they found that this aurora kinase is considered to be a very toxic one. It's actually inhibiting aurora kinase could lead to a lot of toxicity. That is where actually it has given rise to the challenges here. And just be online, please. Sir, వచ్చారండి సార్ నేను ఒక క్లాస్ తీసుకుంటానండి, తర్వాత ఒక హాఫ్ అన్ అవర్లో అక్కడ ఫ్రీ అవుతాను నేను. ఇంతలో మీరు సైట్కి వెళ్ళిపోండి సార్. I'm sorry. Can all of you hear me?
Audible, sir.
Yeah, thank you. So now, so this is the challenge. So what we are finding is the compound 3 is not only inhibiting what is required to be inhibited, but it is also inhibiting some kinases which are actually toxic. Then what are the challenges, what are the options we have? So when we looked at this structure and when we looked at the patent, so there is something called selectivity issues are there, and also potencies, of course 18 and 18 nanomolar, but for a molecule to go to clinics, it's better, always is a better. And more than anything else is, the molecules have to be having patentability. So what we found in the whole structure, so there is one area where actually if we can substitute, we predicted that it can improve the selectivity as well as the potency. So that is one actually we thought. So among this is again, this is the insight which we got from the artificial, that computational tools. When we looked at this molecule and tried to substitute at different positions and try to screen all the molecules against various targets, what we found is this substitution at this position is going to be beneficial. Then what we have done is say for example it resulted into making a molecule like this where we are having a pyrrole substitution here. So this actually made a big difference because what happens is this retained the activity, and at the same time when we made another molecule like a compound 5 wherein substitution like this when we had at this position, this is also very good activity. Then when we substituted with a difluorophenyl, so this also gave rise to very potent. If you really see from 18 nanomolar, if you see this is a 2.5 nanomolar and this is a 4 nanomolar. So if you see this whole success, if you see this, the substitution at this position, it has improved the activity. And also at the same time, what we have also found is this molecule is found to be having a good effect on the selectivity. How it is? If you really see here, this is the data, but we have a selectivity profile. So if you see the TK, which is which is actually at a which is a standard drug. If you see TK, actually it is inhibiting not only FGFR, but JAK2, CSARC, Aurora kinase. Then compound 3, it is definitely affecting the Aurora kinase, and in addition to that, it also has some effect on the JAK2 and CSARC. Then it is having some effect on the CMET, RON. So all this actually it is having some effect. But whereas when you see compound 4 and compound 5, which actually came as the 2.5 nanomolar, 4 nanomolar, if you see here, there is a great difference. If you see here, the Aurora kinase from 69 it became 26, from 27 it became 5, 30 it became of course this one, and then if you see here, if you really see most of the places where actually it has the improved the selectivity. It is actually it has reduced its affinity towards the other kinases, and then it is more acting on the FGFRs. So this is something which is very important to note, and also very significantly when when we do this, this is only a few cell lines, few cancer targets which we had in house. But when we tested against more than 20 kinases, so here actually if you see here, this is a very interesting thing. Then compound 3, compound 5, then TKI. The compound 3 is the one which is actually a unsubstituted one. Compound 5 is where we are having a substitution here. TK is what is called as a Novartis compound. It's a kinase. It is a multi-kinase inhibitor. If you see here, the compound 3 is inhibiting wherever we have the green. So that means it is having significant inhibitions. So you are having able, you are having c-KIT, you are having FGFR1, which is actually desired. Then also you are having MLK1, and also there is another another kinase called MuSK kinase. Then you are having a PDGFR. So if you see here, even though it is having a good effect on some of these inhibitors like FGFR1, which is required. In addition to that, it is also having a another kinase called MuSK kinase. Usually MuSK kinase inhibitors are actually it's related to leading lot of the muscle cancers. The cancers related to the muscles. That is actually caused by MuSK inhibition. And actually inhibiting MuSK kinase is something which is undesired. And if you really then what we have seen is compound 5. You see here from 32 it became 5. And then if you see here ABL1 it has reduced. If you see CK it is reduced. And if only FGFR1 it has done. And if you really see here, so this is something which is very much encouraging for us, because what happened is at 100 nanomolar and you are able to bring out lot of selectivity and this is actually coming from this. And actually I will rationalize this with using and all this was done not because of and the challenge here is supposing if I have a molecule like this and if I want to go on substituting, I can put one substitution here, I can put one substitution here, one here, one here, one here, one. So all these are possible. But in order to prioritize which one molecule to make, that is where we had a lot of the designed molecules which are screened against these kinases like a Musk kinase, Aurora kinase. Then we have we have selected only some of the substituted molecules at this position, and we were quite successful in doing that. And if you see here, it is 100 nanomolar, at 1 micromolar also it has reduced. Even at 1 micromolar, if you see Musk kinase, actually it's only 1.87. So that means it's quite significant. So what we are able to get the selectivity. Then you see, and we have tried to rationalize. Then how do we make this molecule? How to work on these molecules? Then why this selectivity is coming. So what we, and just to summarize kind of the one which we have seen here. So you see here, then what we are having compound 3. So the compound 3 is actually, and compound 3 is in blue. So if you see this, there's a blue, and also you are having a compound 5 which is green. So if you see here green, so you see there is a phenyl substitution, parafluoro phenyl substitution which I am putting here. And if you see gray blue, this is actually stopping here. And actually what we are having here is actually a P loop of the Musk KD homology model. And actually, this is something, if you really see here, this blue one, if you really see here, this is something which is actually a P loop, and this P loop of Musk KD is actually having a very, it is occupying this space. And when this position is occupied, then what happens is when you are having a ligand, which is actually having a substitution here, obviously this sterical clash will not allow any substitution to be happening here, or any compound with a substitution here cannot bind to Musk KD. But on the other hand, when we are having a blue compound where we do not have a substitution, naturally it is stopping at this. So naturally this place is unoccupied. So when it is unoccupied, naturally it will be this can as well bind to Musk kinase. So this is how we can rationalize how we can do it. I think this is an example which I want to show how we are able to make a molecule like we are able to put a substitution in the desired place so that we are able to get a desired selectivity. So I think this is one example which I could show, wherein how we are able to utilize the computational tools like using the molecular modeling techniques, how we can bring about the selectivity. So at this, now we are having a molecule like this. R2 we are having substituted phenyls, R5 or 6 moment heterocyclics, and then what we have done is we went on to make other parameters like N-methyl pyrazole to other heterocycles, N-acetyl to sulfonamides, ureas, other related. So the other traditional medicinal chemistry approaches have been applied. And then we made a number of molecules, especially acetyl is replaced with the sulfonamides. Then among this, what we found is this molecule compound 10. When cyclopropyl sulfonamide is made, it's having FGFR proline inhibitors, and then it is having a good metabolic stability. Then if you are having a inhibition in the cell lines, it is showing around 21 nanomolar. Then this is how actually we can optimize it. So compound 5, which is having an parafluorophenyl, so it is changed to the compound of 10, where we are having a cyclopropyl sulfonamide. At this stage, I also, even though it is not completely directly related to any the topic which we are discussing, but there's a very interesting observation what we found. So you are having a N-methyl pyrazole, which is directly connected with the CC bond, and what we also did is we also made a CN bond, like a CN bond here. When we made a CN bond, like sulfonamides and compound 13 and 14, what we found is this molecule is also active. It's like a 10 nanomolar, 16 nanomolar. But what we found is this molecule, along with compound 5, compound 10, So when we tested them in vivo models, what we found is these two molecules, they have actually shown very profound toxicity in the xenograft. That means, in the animals. So that is something, which is very, very important to note, that, we do not know even to this day why this toxicity for these particular molecules have come. Whereas with CN bond, but whereas, the CC bond, it was quite safe, you know. So that is why, based on all these studies, compound 10 emerged as the lead compound. Then, this is the profile of the molecules where we are having compound 10. Then, you are having, it is on the various FGFRs, this molecule is quite active. And also, I think in the, also very important to note that, the molecules, whenever we think of a kinase inhibitors, also we have to say there is a mutations are possible. So we have also taken a mutated FGFR1, which is called Phi. This is all molecules against that also. Then cell-based studies were done, like, different cells which are in which FGFR is overexpressed. So these are all also in the good range. Then solubility, then MLM stability was done. Then PKPD studies were done. Then found that, the oral bioavailability is almost like a 60%, which is quite good for a drug candidate to forward. Then, we compared with various drug candidates, like, the ones which are actually from the Novartis, then AstraZeneca, and again one more Novartis compound, Eli Lilly. So these compounds were all at that time we have come and we compared with them. So the molecules are quite comparable with the standard the molecules in the market.
Deputy Secretary,
So
National State Board of Technical Education, Mumbai.
and we are having, we found that, since we are the whole project is on bringing the selectivity, so we actually
Some of them
screened the molecules
during
more
field
than
work
300 kinases.
completed time bonded
And these 300 kinases, actually what we are having is we found that the molecules are more specific than the reference compounds. So whereas the Novartis compound is it is hitting more than 95 kinases.
pioneering designing these things.
I think there is some disturbance. Hello, can you hear me?
Yes, sir. Yes, sir.
Yeah, I can proceed.
Yes, sir.
And
Yes,
then
sir.
where
You guys,
AstraZeneca is
you guys.
inhibiting more than 53 kinases. The compound 10, actually, if you see the number, it is hitting only 20 kinases. So that means if it is more selective than the competitor molecules we had, then it is to have less kinase based adverse effects. Then, it became a very good molecule in the xenograft studies, with a clear role in different phases of the cell cycle. So then we took the molecule in a H1581 xenograft model, then which is actually highly FGFR dependent and including FGFR amplification. It's in the nude mice. Then compound 10 we have done it at different concentrations. Then so we found that the molecule actually has having a this is you are having a control vehicle. This then we are having compound 10 at 10 macromolar, then then 50 macromolar, 50 mg, then we are having at 30 mg. So this is a once a day treatment, and this is a twice a day treatment. And we see here the one which is actually that yellow color, so this 30 mg kg twice daily. So this is actually it has become a negative. Virus. It has actually negative growth is there, whereas the and then when you take this green color, so this is actually coming here. So this is at a 10 mg per day. So it is actually compared with the vehicle, it is quite less. Then when you have a compound, 50 mg per kg, so this actually is on par with AstraZeneca compound. AstraZeneca it's 12.5 mg, with this is a 50 mg. Then you are having a compound at 30 mg. So this actually, if you really see, there is a dose dependent decrease or dose dependent inhibition of the activities. So then you are having a dose dependent and significantly inhibited the tumor growth. With the dose 50 mg per kg, the efficacy was similar to AstraZeneca. So this is something which is a very significant one. Then of course, we made the molecule in a discovery route. Then we have also optimized the synthetic steps. This is overall it had 11 steps. Then we reduced that to from 11 to 8 steps. This actually we avoided this synthesis. Then we went for a number of patent applications. Then we had more than 16, 15 patents, and then the molecule was out licensed to some company, and then that was there with it is in the phase 1. It passed at the time, and phase 2, but now it's currently in the phase 3. So this is something which I wanted to show as a case study, wherein a good combination of medicinal chemistry along with artificial intelligence tools actually can give rise to a very successful drug. So maybe I just Lakshman, I can just take one.
Okay, sir. Okay, sir.
Okay, sorry. So at this stage, do anyone has any questions to answer? This is one of the major case study which I wanted to show. Or we can take the questions at the end. So we will proceed now, and then maybe I will take the questions at the end. Okay. So this is something which I wanted to show as a example where we are having if you really see this case study, we had a good biology, we had a good chemistry, then we had a good scale up, then we also had a important component of the computational chemistry, which actually played a very significant role in identifying a compound which is a highly selective and efficacious compound. Okay. So this compound, when we have done it, is it acted in different tumors, like a solid tumors. Then you have then. So now we go to another case study. So this case study is actually the previous. I think I should say that the case study which I talked about, this kinase inhibitors, is now done in the industry called Origin Discovery Technologies. It is not from the NIPER. But from the NIPER, actually, we had a small study which is done, which is actually a structure-based design of new thionopyrimidine derivatives as antibacterial agents. So here, if you see here, so these molecules, this thionopyrimidines, so thionopyrimidines, I think all of you know that this is a pyrimidine molecule, and this is a thiophene molecules. When this thiophene and pyrimidine are combined together, that's called thionopyrimidine molecule. And these thionopyrimidines, there are in the literature, there are many examples, where we have this is known to be having a good antibacterial activity, like and Staphylococcus aureus, and also the compound known to be inhibitors, what is called as TRMD inhibitors. So the compounds are not TRMD inhibitors. So based on this, what we have generated is we have generated a large number of thionopyrimidine molecules, like what we have shown here. So a number of molecules we have actually designed, and also based on the structures of some of these molecules, okay, this is also thionopyrimidine, wherein we are having a MIC of 0.78 to 12 micrograms on Mycobacterium tuberculosis. So based on these structures which are already available in literature, and some of the other structures, closely related structures. We designed a number of molecules like this. So these molecules, what we have done is we made 5 sets of almost like a 200 molecules which we have designed. So these are only designing molecules. We have done only molecular designing, and these molecular designing we have taken what is the TRMD, which is called tRNA methyl transferase. So this is a target which is a very promising target in the antimicrobial target. For example, both on the Staphylococcus aureus as well as on the Mycobacterium tuberculosis. So this we have taken as the therapeutic target. So this target, what it does is actually this enzyme is responsible for methylation of the guanosine. So like this, we are having NH, wherein we are having TRMD. So this actually gives rise to methylation. So this is distinct from human counterpart TRM5, a priority antimicrobial target. So then you are having the target, then you are having tRNA, which actually transfers methyl. This methylation happens. So you are having a molecular designs, then you are having we have designed, we have actually done a virtual screening of all the 200 compounds against this target. Then we found that there are 3 sets of molecules, 30 compounds. We had done the molecular, out of which we have synthesized around 22 molecules out of these 30 compounds. And these compounds, we have evaluated them for their antibacterial as well as antimycobacterial activity. Then we have also done the 3D QSAR admin studies. So this is something which I wanted to show you as an example how a virtual screening from 200 compounds we have reduced to 30. So these advantages of this is when you are having a large number of designed compounds, and when your capability does not, it is not possible for you to synthesize all of the compounds. So then you can prioritize based on what is called virtual screening. So the molecules which we have done, the 30 molecules, we have synthesized it using the cyclohexanone as the starting point, and then thionopyrimidine using a very standard protocols. Then this is also another standard protocol where you are having a pyrimidine, so you are having a n protected ones. Then these molecules, what we can show you, some of the molecules we have screened them. So what is very important to note is these molecules, when they are tested against various antibacterial strains, like both gram positive and gram negative bacteria up to here, and the escape pathogens, and also Mycobacterium tuberculosis. If we see here, these molecules, some of them which are highlighted in red, 13B, 18A, 18B, 18C, and here, these molecules, they selectively bind to Mycobacterium tuberculosis, like a 16 micromolar, 16, and 16, and 32. And this is something quite significant because this is highly selective. So maybe from the potency point of view, still there may be a lot of requirement for optimization. But nevertheless, we got a good starting point because of MSE of 16 micromolar. Then these molecules were, tested when we have this binding energies of these molecules were calculated, and we found that the molecules are having a very good potential, like very good binding energies. And then these molecules, when they are superpositions of molecules at the binding pocket of the TRMD, so the TRMD is the target. So the TRMD target is actually binding very well. Then we had based on this TRMD. So this molecule, this work actually has been published in Journal of Molecular Structure, wherein we have 3 different sets of 22 compounds, and the antimicrobial activities were tested against. What we found is, even though we have tested against various bacterial as well as mycobacterial strains, so this molecule has shown activity against this Mycobacterium tuberculosis. And then so this is how we could do, we could apply a number of these molecule, these techniques into our drug discovery process. So these are the 2 case studies which I wanted to share with all of you. And now If there are any questions, I will be happy to take.
If participants have any queries, please ask to sir. You can also post your queries in Dropbox also.
And even if there are no questions immediately, so I can be contacted by email, then I will be able to clarify the questions even offline.
Okay, thank you so much, sir, for this informative section. Now it's my pleasure to present an e-certificate to you, sir.
Thank you.
1 minute, sir. Sir, please accept it, sir.
Thank you.
Okay, thank you, sir. Assessment and feedback links are posted in the chat box. Please fill the
Sure.
forms.
Yeah.
So this is the end of the section today. Please rejoin the section on tomorrow at sharp 9:30 a.m. Thank you so much, sir.
Thank you. Thank you. Yeah.
Sir, excuse me.
Yeah.
Okay.
Sir, one small doubt, sir. Sir,
Yeah. Please
sir,
go ahead, madam.
after doing molecular docking, if we go for molecular dynamics also, being pharma people, we don't know how to interpret those results actually.
As per actually, see what happens is whenever you do the docking, it has to be followed by the molecular simulation studies, because what happens is the simulation study, because the molecules are not always rigid and the ligands and the receptors are not always rigid, so they will always be in constant motion. For that, actually, we do the simulation studies, because we also, even though I have not presented here, for many of our
Yes.
docking studies, virtual screening, we have to follow up with the simulation studies.
Yes,
So
sir.
if
Sir,
you
do we have
happen,
any courses available, or any online courses or any material through which if we can go and we can learn it? I mean, are there any learning tools for this?
as such, I have not gone through any of the courses, but if I can come across, I will let you know. But otherwise, what we
Okay, sir.
normally use is we use the Gaussian software. We have been using it.
Okay,
If
sir.
you want some any help, I can help you out with one of my students who is having a good experience in that.
Okay, okay. Thank you so much, sir. Thank you, sir. Surely we'll
Okay.
contact you. Thank you, sir.
Thank you. Thank you.
Okay, thank you, sir.
Thank
Have
you.
a nice day,
Thank you.
sir.
Thank you. Thank you.
Okay, sir. Bye.